And so our next speaker is Paul DeMott from Colorado State University.
All right, thanks. So I think I get to talk about the messiest topic here. There are a lot of people working on this right now in the world, as it turns out. It's a vibrant community. It's changing. There's papers coming out every week. I can't hope to represent that all here. They gave me 9 hours to talk about this topic in a summer school in China last year, which I really appreciated. I'll see what I can do in 30 minutes here. Okay, so I'm going to dive right into it and start with a figure that came out of a paper that I published in 2010. I'm not going to go through the details. You know what all is going on up here deals with clouds and the radiative impacts. That's the next step. I think we need to focus a significant amount of energy on the lower part in defining what are the specific emissions of what we're going to call ice-nucleating particles. This is a sort of a new term. You're going to hear me say it a lot, and I'll try to explain it along the way and why we're using that terminology. We've known for a long time about mineral dust, their role, and that's just been reiterated over and over, and you're going to hear it through my talk. We want to know about all these other types of emissions, from biomass burning, from pollution, from oceans. This is something that I ignored greatly earlier in my career, but the fact is these are a massive part of the surface of the Earth. And it must be the case that they can dominate the emissions and the effects on even cold clouds some places on Earth. And we know about that, we know about the biases in the radiative balance over the southern oceans. One of the potential contributors to that, although there could be many reasons, and people in this audience know better than I, dynamically and so forth, why that could be happening and microphysically, the fact is that aerosol emissions could be part of the story. Also, biochemistry happens in oceans. There's blooms happening, and we want to know if that changes the input of ice-nucleating particles. So a lot of my emphasis today is going to be on both dust and emissions from the oceans, and that's at the very end. So when I started to put this together, I had to ask myself, well, what are the big questions? Because I can, on a daily basis, have my head buried in details of this very complicated subject, and you may all have different answers, but I decided I'd focus on a couple of things. The first one being, can we effectively measure them? That's the biggest question I get from people, and I hear from people, is whether we can even measure them. And I think that I can give you a strongly qualified yes, and I'm going to try to support that today. This is a big question. Do they equal ice and clouds? Well, the better question might be, when should they? I was glad to hear you talk about secondary ice processes. It's such an important factor, and so there's only a certain variety of clouds that you can go out and answer this question directly or do your experiment very specifically to do that. So I'm going to spend a lot of time on, do we understand what aerosols are important? And you can see I'm going to emphasize mineral dust, the potential for vast potential for biological processes to contribute, but not yet verified, and down to what I'm going to call subparticle sizes. So that means that the particle you're dealing with, that we're calling the ice nuclei, it's not the entire particle. It could be some small component of it, and there's lots of evidence for that now. There should be a strong contrast, I believe, in land versus ocean inputs of ice-nucleating particles, and that's sort of a low-hanging fruit that I think we should go after. And there's a lot of other questions that are extremely important that I'm not going to have any time to talk about today. Okay, so what do we have to measure? Even conceptually, it's complicated, and this is very simple, and you've all seen this probably in textbooks about the different processes that by which you can nucleate ice. And now we're calling them different things. If you can be more specific about what they are, that we're asking people to be that way. That's what this paper is all about. If you know it's a molecule that's causing it, say so if you think you can. Now, what the fact is, I don't have time to prove this, but probably the most important process is this immersion freezing process for many mixed phase clouds. I'm not talking about ones that are very cold down near cirrus cloud temperatures, as Thanos was talking about earlier. What we need to provide, ultimately, is some way to quantify the ice-nucleating potential of the particles feeding that. And you can do that deterministically, which is the most common way, just give an ice-nucleating particle spectrum, number concentrations, or related to the surface area of a certain classes of particles, and it's just a function of temperature. Really, though, we understand that nucleation is stochastic, and some people go to the extent of trying to put all these results into the framework, say, of classical theory and do this in a manner that can represent the vast array of contributions that can come from different aerosol particles. The important thing to know is that ice nucleating particles are but a very small fraction and at very small numbers compared to the total aerosol population typically. What is the space we have to measure? And to do that, I'll just show some results that many people show from Al Cooper back in the 1980s of measurements in clouds where he thought, at least, that secondary ice generation wasn't occurring. And you can see that they span many orders of magnitude, and we're not even down to temperatures still where homogeneous freezing is not even happening. And the variability is tremendous, okay? So we need to understand this and explain it. And we don't really have measurements much like this available from recent projects, and that would be very useful. So the way we do it is with a variety of instrumentation that I'm not going to go through in detail. I'll point out that we're in the midst of what's called the 5th International Ice Nucleation Workshop, a collaborative effort between people in the United States and Europe and other places. And we have a laboratory session, we have a field session, we have a session for people to show that they know how to measure the compositions of ice nuclei. But in any case, typically we will have multiple measurements, and I'm going to promote that we have multiple measurements in any field effort. This might include a real-time instrument, a continuous flow diffusion chamber that can activate the particles and then potentially isolate them as well after freezing. But that has a flow rate of about a liter and a half a minute. We're not going to measure concentrations of some numbers per cubic meter with that method, so we have to sample in bulk onto filters, into liquid, and so forth, and do classical immersion freezing methods. And now we have to show that those give comparable results. Some cases they do, in some cases not, but I'm going to jump into that in just a second. Well, this is kind of scary, but not many people have been out making measurements in the atmosphere for the last 20 years or so, and this is where we made our measurements. Well, we're not covering a lot of the globe, right? So if we added in offline measurements where samples are collected, that would add more. We'd have one down here in the Amazon, we'd have some in Europe and so forth, but we'd still leave huge areas closed. So another scary thought, have you ever thought about the sample volumes we've collected? And I did this. We had 14 aircraft campaigns and 11 ground-based ones, typical amount of good quality data we get from each. Work out the numbers and you get about 90 cubic meters of air sampled. Has anybody been to the AIDA chamber in Germany? That's about the volume of that chamber. That's what we've collected on aircraft flights and in the ambient atmosphere with continuous flow chambers. Now the situation for offline samples is better. And even in just the last 4 years, we've collected about 16 cubic meters by that method. And we have to use that method if we're going to access, like I said, these lower number concentrations. Of course, when we go out there, we're not always just measuring the ice nucleating particle number concentrations. We're trying to learn more than that. We'd like to collect the residual particles. We'd like to sample off multiple inlets, a CVI, getting cloud residuals, ambient air. Collect, like I said, the residuals of the activated ice nuclei. And do that and send it to specialized instruments. Maybe we want to know if there's black carbon are involved or biological particles. Or we want to measure their mass spectral compositions. That requires sitting at one set of conditions for a long time. Field campaigns are not designed for that, okay? People want to be in one area, they want to be in the cloud in the next minute. We could use some specialized programs, so I would promote that. Additionally, you talked about inlet issues. We have the same problems when we try to sample from aircraft. We have to dry the air before we send it into a cold instrument like this, a flow instrument. And sometimes we want to purposely remove water, heat the particles to look at the effect on biological particles specifically. So how are we doing? This is an example of just sampling one dust in the laboratory by 3 different methods. The gray line is results from a classical immersion freezing method. We have the AIDA chamber results with most of the colored red and black data points. This is for K feldspar, and a couple of CFDC data points where we sat at one set of conditions for a long time. I'd say that's not too bad right there. There's a broader story about K feldspar, but I'm not going to have the chance to go into today. That's not so good. But what about ambient aerosol measurements? So here's a combination, and this was startling to me. We did this blind. We had Marcus Petter's group from NC State come out and join us. Alan Bertram's group was there, but his data weren't on this day. We collected filters, we collected particles into liquid and biosamplers. We ran the continuous flow chamber. Since the ion concentration at minus 20 was only on the order of 1 per liter, we had to sit there for about 4 hours to get really, really good statistics. But the amazing thing was this immersion freezing method puts emulsions into oils. This one distributes into wells that are, say, 20 to 50 microliter. They agreed tremendously well, and the two different ways of collecting agreed very well. And we've got ice nucleating spectra over 7 or 8 orders of magnitude. This is the kind of data that we need and have the belief that they're actually comparable. Now, to get these kind of data with, say, this immersion freezing method versus the continuous flow chamber, we'd have to operate for these 4 to 8 hour periods on individual days at different temperatures. And here what you see is that we're segregating a little bit where the immersion freezing data is actually computing more ice nuclei than the continuous flow chamber. Well, we've always understood that we might have biases, and one of them could particularly be the size of ice nuclei. We can only bring in a certain size into the flow chamber. We cut it off typically at 2.5 microns. So I'll try to put the light there and show you. This is a method that uses a Moody sampler and gets samples that are then processed for immersion freezing and finds out the size of the ice nuclei. Well, we're missing fully 50% of them due to our cutoff of the entry of aerosols into the continuous flow instrument. So this is one of the biases we need to be concerned about and why we need additionally measurements like this if we're going to characterize the entire distribution that's available. Okay, I'm going to step now into what we know about the sources then, using these measurements and assuming that, that they're meaningful. And so these are the types that I'm going to hit on a little bit today. Okay, I'm not even going to begin to try to explain this one. This would be a contentious discussion, actually, because there are different ways of measuring the composition of ice nucleating particles with different mass spectrometers in different situations, collecting with CVIs or collecting the activated ice nuclei, people collecting ice crystals and looking at their cores. Instead of talking about the parts of this that might be discrepant, I'm going to focus on the parts that are common. The one common element is the role of mineral dusts, whether it's just them alone or the things that come with them. And fully 50% or more in all of these cases are things like mineral dusts, metal oxides, and so forth. What's absent, or what's mostly absent? What's mostly absent is black carbon, a little sliver there, hardly visible here. We have carbonaceous aerosols here, but that's all carbonaceous aerosols. And the sliver that specifically has a morphology of soot would make up maybe 6-10% maximum of that. We know they're there, but that's not the major contributor. What also is missing, largely, and hard to really identify, is pollution. Now, a different picture would be given by Daniel Knopf and colleagues in some recent results of aerosols sampled right around this region here, where we have this mix of urban and marine aerosols, and he's basically promoting that they're all ice nuclei under some conditions. But they're suspiciously absent in a lot of these data that we have collected, admittedly very little, as it turns out. One nice thing about mineral dust is that it appears in the lab, and this remains to be challenged by other people, that they can somewhat be unified in their behavior in terms of the active site density, the number of nuclei per meter squared of surface area for different dusts from the Canary Islands, from the Sahara, from Asian regions, and so forth, from Israel. They can somewhat be unified by this parameter, and that could partly be understood by the role of certain feldspar minerals. At least that's what's been promoted by Ben Murray's group in this article in Nature. We saw the same thing now that when we've compared both laboratory data with field data on what we could just characterize as being, say, Saharan dust or Asian dust. And so in this paper out recently, we see similar behaviors, whether they're generated in the lab or the field. And so when I saw Thanos's results that showed that the lab data used in the models didn't necessarily agree, I think I guess we have some work to do on the parameterization side of things, it seems like, and getting those into the models really well. So that's the good story about mineral dust. We can use that information and look at field data. This was collected at the DOE SGP site to see that, well, on average, the particles there over a month period did look kind of like mineral dust. But this is in the spring in the center of U.S., so why do we think they should look specifically like mineral dust rather than soil dust? And that's one thing, I think that's what I'm coming to next. And that is, what are we missing? What we're missing is we don't understand the contributions of biological particles. They're hard to measure and identify. There are lots of organic particles in soils we're identifying in laboratory studies that are ice nucleation active, and they may have extremely small sizes. So in the Beacon study, there were a number of papers that emphasized the fact that after rain events, we'd have bursts of ice nucleating particles produced, and these were, in fact, in line with the production of fluorescing biological aerosol particles. Tobo et al. From our group said that you could unify these, in fact, relate the ice nuclei to fluorescent biological particles. It was a strong relation at the warmest temperatures where they appear to dominate, less so at low temperatures where minerals also come into play. Well, Mason et al. From the Bertram group in British Columbia has a paper out just right now in ACPD, and it looks at the same relationships along the coast of Vancouver Island. And what they found was when they used the Tobo et al. Relation that unified the measurements the best, relating to the bioaerosols, then they did not get results that were equivalent to theirs. It had somewhat the same slope, but they were over-predicting ice nuclei by about a factor of 10. And it's possible that this is because of the large marine aerosol influence there and the fact that the biological particles from marine aerosols are not necessarily the same as the ones in the nearby forests that are both potentially affecting that site. So, but in any case, we don't know yet if we can unify bioaerosols and have fluorescent bioaerosol ice nuclei in a model. There's a vast amount of literature and a lot of people in biological sciences involved in looking at ice nucleating bacteria. So this is these are some of the most active things on Earth to make ice potentially in clouds. But what Tom Hill has done in our group, he's a microbiologist, and he's developed quantitative PCR tests to actually enumerate the biological ice nucleating particles. And he can identify through the specific genes and their copies the whole array that come from bacteria specifically. And so he finds tons of them on plants, of course. In precipitation, though, specifically in snow, when he looks at the numbers in snow, they cannot explain anywhere near, by orders of magnitude, the number of ice nucleating particles measured per volume of the water from snow. In fact, they don't even measure what happens when you heat the samples up to a temperature that would destroy proteinaceous matter. So there's other biological organisms responsible for a lot of the ice nucleation in those samples, but it's not these ice nucleating bacteria specifically. Similarly, when we make measurements, say over the Great Plains of the US, we find again a heat sensitivity which identifying proteinaceous ice nucleating particles, but they're not the ice nucleating bacteria that we are all familiar with. In some cases they can be, those do explain, this is how many he counted, and we put that at a temperature of -12 because that's where a single ice nucleating gene will freeze a droplet. And this is in a corn harvest though, so it's only in these highly perturbed situations that you get correspondence between the ice nucleating bacteria and the ice nuclei. So there's this other latent population that's unidentified as yet. Some of that may be coming from soils because we found that this is a typical desert dust, and when you heat it, when you treat it with hydrogen peroxide and then crumble it up again and generate it again, you really get the same result. There's very little biological material in it. When you do that with a arable soil, this one from a field in Wyoming, you see that there's a very strong signature toward more organic ice nuclei at the warmer temperatures and then only more mineral as you get to the very low end. So arable soils are quite distinct from mineral soils in their potential contributions if you have ablation of soils from land use changes. This just extends that same plot using the two different methods now, the continuous flow chamber and the immersion freezing spectrum, to show that they populate, they're describing the same things, except we have a hump, and these truly are the proteinaceous ice nuclei here. These other organic ones, we're not sure. So that's the big question. What could they possibly be? And one source are fungal INP in the soils. Those can be removed through the use of something called papain and through enzymatic treatment. And that removal equals the removal you get from heating them up to 105 C in liquid. So we think that those are strong contributors in these soils. But O'Sullivan et al. Have pointed out that you don't need the whole fungi, that it's actually very small elements, what they're calling biological nano-IMPs, that are actually responsible for the ice nucleation. I'm going to skip on from this. Just to say a word about biomass burning. There's a lot of rich literature here over the last few years, but the fact is, in something like prescribed burning, the fire doesn't have great power. You can have an input of a lot of particles, these the ones bigger than a half a micron, and have only modest changes, an order of magnitude over orders of magnitude change in the particle inputs. Whereas wildfires, they do a lot. They create ash. They can kick up soils. And I think that's the reason why we saw that the wildfires looked more like dust particles than they did the smoke that we produce from prescribed burns. So this bears a lot of additional investigation. I'm not going to talk much about pollution particles. There's been a few papers, at least, that give inferences to this. I think the jury is out, and the ice nucleation data directly available is only from China and has never been reproduced. We have a lot of work to do there. So I'm going to end talking about marine aerosols. And a lot of this work was done at the Center for Aerosol Impacts on Climate and the Environment at UCSD and NSF Chemistry Center. And a lot of it was done out in the field over the recent few years, making collections on ships and other places. This is just a cartoon to show that you do get sources of ice nuclei from the oceans. They change with blooms. They can come from purely biological particles, from coatings on sea salt particles, from small things that we can hardly identify, and in fact from monolayers of fatty acids even at sufficiently low temperatures. So here's a compilation of measurements, some taken in the field in these different locations and other groups even, from Alan Bertram's group and ours, and ICE-T experiment, MAGIC studies going back and forth from Honolulu to LA. The case measurements, those are in red from different methods. In blue are the field measurements. Everything's normalized to 150 particles per cubic centimeter. And interestingly, we create the same apparent distribution, which is nice. It means that we're doing a good job of generating the aerosols in the laboratory. Also interesting, we span the variability that has been seen over oceans in the southern hemisphere, just from these samples off the Scripps Pier. These measurements have been questioned many times, but I think they deserve closer inspection and repeat. This is a distinctly different emissions than over lands, and we should expect so. So these are the data from our high plain studies here. They separate themselves, much more potent IN from arable land sources. But when you have blooms in the ocean, you can have generation of ice nucleating particles in different temperature regimes that are in fact different types of particles, and I won't have much time to go into that today. Here's the same data now normalized by surface area, and this is still a work in progress actually. Getting the dry surface area distributions from the field data is quite a task. And then now we see maybe some segregation with the lab data, which I'm not going to try to explain today. But it is somewhat the same spectrum still, and these are for arable dust, mineral dust, and here's some diatoms as interpreted by Ben Murray's group from data by the Knopf group. And that's interesting because in measurements at minus 30, in these case studies, we isolated ice nuclei for the first time for Raman spectroscopy studies. And at that temperature, by comparing reference spectrum to the spectrum from the ice nucleating particles, fully 50% of them appeared to be diatoms and biofilm. So that was the first actual collection of these particles and identification of these different sources. Like I said, though, there's lots of other sources that we now understand, and we're doing isolated laboratory studies on these different types. I don't have time to go through the details of this, but I want to focus on one part of it. We can do studies of filtered seawater and heated seawater to understand the sizes of the elements in it. But here's a filtering at 20 nanometers, And I want you to notice that there are contributions from units in seawater as small as 20 nanometers as isonucleating particles. So that's a significant challenge to understand what those are. Finally, we've gone back to making these kinds of measurements where you connect a continuous flow chamber. We enhance the aerosols with a pre-concentrator to try to get better statistics, such that what we did in about 5 days here on the coast of California was what took us almost 2 months to collect the same number of particle statistics on Storm Peak back when we did this back in 2003 or whenever that was now. So what we see is that it gives us insight into the special character of the ice-nucleating particles. This one is an aged sea spray aerosol that was an ice-nucleating particle at about -30. The aerosol was dominated by sea spray, both aged and fresh, but when you process them, especially at this temperature, while you still see that 50% of the inputs are coming from the sea spray due to its dominance, small categories such as dust or dust and biological particles mixed stand out because they're highly efficient ice nucleators in this environment. So you can imagine if you're far away from those sources, these things dominate, but when you get near the land, you have to consider all these. So here's my summary and outlook, trying to stay on time here. It's a daunting measurement challenge, and the parameterization challenge may be as large if we have so many sources of so many unique sizes and compositions. There is widespread use and development of methods for real-time and offline detection of these particles and their sizes and composition. It's happening everywhere. You're going to see lots of things coming out. Question I have, can we ever make a link to remotely sensed quantities? There's been a little bit in the literature on that. We should be able to confirm drastic differences in the sources in certain regions. For example, regions that are dominated by oceanic sources only versus continental ones. That would be a low-hanging fruit kind of thing to go after in a field study. Arable soil contributions are not well constrained right now, nor do we even know what we should be looking for, say, in the mass spectrum. The role and diversity of biogenic ice-nucleating particles is linked into that directly. We don't know much about pollution. We need to sample that specifically in some studies and see if we can understand, because that's the most obvious human impact besides, say, land use changes or changes in temperature affecting biological particles. So I think that's about all I need to say. Thanks.